We now welcome the academic procession. Please be seated and welcome Dean Nerges Mavalvala to the lectern.
I hope the applause is for you, the graduates. Good afternoon, everyone. Good afternoon, especially to our guests of honor, our graduates sitting in the center over here. Good afternoon, parents, friends, and families gathered here to celebrate the MIT graduates of 2024. We have much to celebrate with our graduates and family and friends here today, but I also want to acknowledge that this has been a profound year in which we have seen demonstrations and acts of conscience that echo past eras, like the anti-war and the civil rights movements of the 1960s and 70s, the anti-apartheid movement of the 1980s during my own time in college. Then and now, students across the US and the world are advocating bravely, loudly, and unapologetically for what they feel is right and just. I know this has been a year that has divided our community, our campus community, and caused many pain. In the past few months, every one of us has experienced the challenges of a community divided. Today, however, we must step outside of all of that and honor this moment for our graduates and their guests, every one of them, the ones who could be here and the ones who could not, the ones who are here with families and from far and near, and the ones whose families could not be here to join them. I ask that we focus for this ceremony on what brings us together and not what divides us. And let me say, nothing brings us together in a more celebratory way than celebrating the stunning accomplishments of you, our graduating students. Students, you have made unprecedented scientific discoveries. You have learned and taught. You have earned your place among the most talented scientists in the world. You will make a better world with your science, with your conscience and activism, and most of all, with your empathy and regard for humanity. Never stop being the catalysts for the change you want to see in the world. Families and friends, you may be wondering what kinds of achievements your graduates have made that have earned them the right to strut across this stage in this display of, shall we say, fashion. First and foremost, all our graduates have made indelible marks in their scientific fields, contributing to research that will have lasting impact on the next generation of scientists and indeed on our society. You, our brilliant students, have contributed to our understanding of the progression of diseases like Alzheimer's, Parkinson's, and Huntington's disease. Using genetic, cellular, and systems-level analyses, you have identified targets that could one day lead to treatments. You have identified a new behavior in black holes that reveals exciting possibilities of complex and undiscovered star systems. You have revealed the ancient ability of a clay mineral on the seafloor to sequester carbon over millions of years, likely contributing to the Earth's cooling in the absence of us polluting humans. You have led the largest study of deep neural networks trained to perform auditory tasks, demonstrating their promise as models of human hearing that could be used to develop better hearing aids and cochlear implants. Using machine learning and radio telescope observations, you have detected a previously unknown molecule, 2-methoxyethanol, for the first time, illuminating how stars and planets form. Physicists among you have contributed to the development of a technique to arrange atoms in the closest proximity yet, from 500 nanometers down to 50, potentially unlocking doors to building new quantum materials and computers. Your scientific achievements are part of your legacy, as is your service to your community and to MIT. Your passionate advocacy helps us all strive to become our better selves, whether it's pushing our climate and sustainability efforts, advancing our knowledge in life, health, or quantum sciences, or mathematics, or ensuring that your voices are heard through demonstrations and dialogue. On behalf of the School of Science, the Woods Hole Oceanographic Institution, and all of MIT, we recognize and congratulate you. We celebrate your graduation from MIT, my beloved alma mater, and soon to be yours. Now, graduates, before you cross the stage, I have one last assignment for you, and I know you're tired. You've done many assignments already, but this is my challenge, my charge to you, to think about how you've been supported by your mentors and loved ones and pay it forward. My own trajectory has been a series of mentors who came in many different guises, from teachers and family members to technicians and support staff, and everyone who has made my own career possible. And I hope, as an experimental physicist, and I hope you will benefit from it. You have benefited from that and will do the same. Now, even before you got your fancy degrees, which are here and you will get them, you have mentored others. And now, along with your fancy degrees, you're a little older and hopefully a little wiser and certainly a little bit more tired. You are the newest mentors in your chosen fields. Take up the mantle and be a mentor and pay it forward. This year's commencement speaker is nothing if not a mentor in all her roles as researcher and rabble-rouser. Dr. Lydia Villa-Komaroff has worn many hats, or you might say lab coats, although I learned recently from Lydia that when she was working in the lab as a graduate student, they didn't wear lab coats, but that was the statement of fashion at the time. In her journey from a young person interested in science to a leader in the discovery of the bacteria, discovery that bacteria could be induced to produce insulin, a discovery that has revolutionized biology and biotechnology. Dr. Villa-Komaroff spent her young adult life in New Mexico, immersed in the sciences, learning from her chemist uncle and from her mother, who was interested in botany and went back to school for her undergraduate degree. She attended an NSF summer training program in Texas, performing experiments, and indeed taking them home with her. If you have a chance, ask her about the black and white rats that lived in her neighborhood. Now, that experience influenced her path to enter the University of Washington to study chemistry and physiology. Her first advisor, and I do use that term extremely loosely and even somewhat cringingly, said, "Women don't belong in chemistry." So she switched majors to biology, and fortunately for us all, that led to another formative laboratory experience which solidified her interest in developmental biology. When she moved to the DC area, she transferred to Goucher College, in part because the more research-active Johns Hopkins still was an all-male school. Through connections at Goucher, she landed a summer job at the National Institutes of Health, working in the lab of Dr. Loretta Levy, a microbiologist who introduced Dr. Villa-Komaroff to the concept of molecular biology. It was Dr. Levy who encouraged Dr. Villa-Komaroff to apply to MIT for graduate school to conduct developmental and microbiology research. Here at MIT, she worked in the labs of Professors David Baltimore and Harvey Lodish in the Department of Biology in the School of Science, taking time, in addition to her research studies, to become a founding member of the Society for Advancement of Chicanos and Native Americans in Science, known as SACNAS. She graduated from MIT with her PhD in 1975. She was the third Mexican-American woman in the United States to receive a doctorate degree in the sciences. Not in biology, in all of the natural sciences. I will let Lydia tell you more about the joys of her discoveries in her postdoc years, and they will be prefaced by failures. But ultimately, all these experiences and guidance from all these mentors, she was one of the early group of scientists who first used the ability to join DNA from animals to DNA from bacteria to make useful proteins and study biological processes and human diseases, including diabetes. The watershed discovery of inducing bacteria to produce insulin change the biotechnology industry forever and the lives of millions who rely on insulin to manage their illness. Dr. Villa-Komaroff has had many roles in addition to being a scientist. She served as Vice President for Research at Northwestern University and at the Whitehead Institute for Biomedical Research just next door to us here at MIT. She has been the Chief Scientific Officer and CEO of Cytonome, a company that designs purpose-built cell sorters and is a member of its board of directors. She serves on the boards of Transkaryotic Therapies, later purchased by Shire, and of the American Type Culture Collection, or ATCC, the nonprofit Resource Center and Standards Organization, and leading developer and supplier of authenticated cell lines and microorganisms in the world. She still serves as a senior advisor to SACNAS, inspiring the next generation of Chicanos and Native Americans in science. Her numerous awards and honors include the Hispanic Engineer National Achievement Award, induction into the Hispanic Engineer National Achievement Hall of Fame, National Hispanic Scientist of the Year from the Museum of Science and Industry, and the Women of Distinction Award from the American Association of University Women. Please join me in welcoming Dr. Lydia Villa-Komaroff.
There's another step. Thank you, Dean Malavala. Congratulations, graduates. You have reached a milestone at a particularly difficult time. You've persevered through a pandemic, and it is a very unsettled world. I am deeply honored to be here with you, your families and friends, as well as faculty and administrators to celebrate your accomplishments. I'd like to share some of my family history and a couple of stories about the time after I left MIT, a place I considered at the time, and still do, as graduate school heaven. I felt that way because the faculty, the staff, other students, and postdocs welcomed us as junior partners in important endeavors. Of course, our more experienced colleagues were blunt in letting us know when we were ignorant, missed the mark, or performed below expectations. Then, as now, the bar was high. Each of you has a history that ultimately led you to MIT and to this ceremony today. Cherish that history. Honor the people whose actions resulted in the unique person that you each are. And even if you don't know the details of your story, you're here. And that serves as proof that there is a story and you continue it. My 5 siblings and I all resemble my father, the son of an indigenous Mexican Indian tribe unknown. My mother was a blue-eyed, red-headed, fair-skinned descendant of the Spanish conquistadores from the Basque region of Spain. Over half of my genetic makeup can be traced to Spain, while under 40% is attributable to the indigenous peoples of Mexico. The rest is a sprinkling of Arabic, African, Asian, Jewish, and a sprinkling of Neanderthal throughout. My paternal grandparents, Encarnacion and Luz Villa, came to this country during the Mexican Revolution. My father grew up in a house with no running water and a dirt floor that my grandfather and his older sons built. My father was one of 15, a middle child. Only 10 survived to adulthood, and all worked in the fields as children. My mother's grandfather, Manuel Larrañaga, and his wife, Cleofas, owned a ranch in Arizona. He sent his daughters to get a teaching degree because he didn't want them to marry cowboys. His sons had to stay on the ranch and be cowboys. Both of my parents grew up speaking Spanish, but because my parents had been punished for speaking Spanish in the school playground, they decided that their children would grow up speaking only English. As a result, I felt I was somewhat an outsider in my very large extended family, which turned out to be good preparation to be part of an enterprise where I did not fit the standard model. Towards the end of graduate school, I arranged a postdoc at the institution up the road, and confidently began a project to isolate genes coding for proteins that make up the eggshell of the wild silk moth. Now, this was well before many of the tools of molecular biology were available. There were no kits. Restriction enzymes, as well as many other important reagents, were not commercially available. Phages weren't used as cloning vehicles, and DNA sequencing was in development, and there were no cell phones. Much of the public and some scientists were worried that mixing DNA from different species would result in an uncontrollable plague. So we decided that I would go to Cold Spring Harbor, where recombinant DNA technology could be conducted. Now, I had chosen an important problem. How are genes regulated during development? But it was not clear that it was doable with the model and the tools that I had at hand. I was working alone. I didn't ask anyone for much input. Would it have changed anything? I can't say for sure. But what I know is that I spent a year in Cold Spring Harbor plating out bacteria that I hoped had eggshell genes and waking up day after day to nothing. So I was trying to do not the impossible, but I was trying to do not the improbable, but the impossible. The polyphemus genome, the silk moth genome was too big. The fragments were too big. The efficiency of each step was low. And the bacteria that I had to work with was very badly debilitated that had to be used under stringent conditions. There was no way we could get any clones whatsoever. However, I left Cold Spring Harbor with an aliquot of every enzyme in Rich Roberts' freezer, a very valuable gift. That freezer was the origin of New England Biolabs, still a major supplier of restriction enzymes and other reagents. So I returned to Cambridge. No project, no papers, a valuable set of enzymes, shaken confidence. On my return, Arjun Stratiadis, my colleague in the lab, invited me to join a project he was doing with Wally Gilbert. The objectives of that project were to clone the rat proinsulin gene to achieve expression of the gene in bacteria, and then to do the same with human proinsulin. Rat insulin was first because we couldn't work with human DNA in the United States at that time. The project was obviously important, both scientifically and commercially. The team had a rich source of rat insulin RNA from a pancreatic tumor we got from colleagues at the Joslin Clinic. Members of the team had all the skills, almost all the skills needed, but they still needed someone to put the DNA into a plasmid and put that into the bacteria. Well, while my time at Cold Spring Harbor had been completely unsuccessful in terms of successful experiments and papers, I had learned how to manipulate DNA, and I had learned how to handle that finicky bacterial strain. And I had a treasure trove of enzymes not generally available, and I really didn't want to end my postdoctoral career with an unsuccessful project. Lessons are learned from failure. Don't forget that. Now, I didn't meet Wally until we'd started to get results. Arj talked to Wally all the time. I talked to Arj all the time. The goal was clear. We knew what had to be done, and Wally left us to it. This time, we talked and argued about our approach. When necessary, we called colleagues with special expertise for advice. We thought through every possible difficulty, and we were lucky. Lucky is the wild card in life and science. Cherish it, but don't count on it. Within 6 months, we had an insulin clone that resulted in the production of a minuscule but momentous amount of insulin. When it came to the last goal, human insulin, we had to go to a high containment facility in England. We had to get dressed in hazmat suits. We had to shower in and out of the laboratory. All manipulations had to be done in a glove box, which was a part of the room with thick gloves going into that space through a transparent window. Again, not much worked. So 4 weeks later, we were very frustrated, and we came back empty-handed and knowing that the team at Genentech had beat us to the cloning of human insulin and also to its expression. Nevertheless, we were part of the birth of biotechnology. Biogen had been founded several months before we went to England. I declined a position there because both my graduate work and my postdoc work had been done at some of the best labs in the world, and I wanted to see what I could do on my own. I did, however, become a consultant to Biogen and had a front row seat as Biogen grew over the next 8 years. It was a great tutorial in business and came in handy later when I became chair of the board of TKT. That's a story for another time. I returned from England to a position at UMass Medical School in Worcester as an assistant professor and set out to build a career. In an academic position in a research institution, the key measure of success is research, and research productivity is measured by grants and papers published. I knew this. Nevertheless, I took on too many service jobs, extra teaching, more students, many committees. I didn't focus on my research plan, and so, as a result, the tenure process, which in academia is always never fun, was particularly painful for me, and it was a squeaker. I got tenure and left UMass 1 year later to accept a non-tenured position at Harvard Medical School and Children's Hospital. My reasoning at the time was that I would go to a more rigorous institution. Clearly, I hadn't yet realized that one's research depends on one's own efforts with your colleagues, as long as the institution has reasonable resources available to you. My move was the right one for me, but I made it for the wrong reasons. So you can do the right thing for the wrong reasons. I was at Children's Hospital and Harvard Medical School for over almost a decade. I had great colleagues, students, and postdocs, and we published on a wide variety of subjects. Many of those papers continue to be published in the current literature. We used the tools of molecular biology to investigate the development of vision, the effect of amyloid peptide on neuronal cells, secretion of insulin, the DNA sequence of several genes, development in the human brain, and the role of a couple of insulin-like growth factors. Each question was interesting and important. However, focusing on so many different topics and not following up on initial findings was not the best way to gain professional recognition. After 10 years, I decided it was time to explore my options. Initially, I thought I'd go back to the lab full-time because I really loved it. But after consulting with many people, it became apparent that I had strong administrative skills, so I began to explore those options. I moved to Northwestern University as associate DPR, and 2 years later, after a nationwide search, I was appointed vice president. It was a wonderful time to be at Northwestern. There were sufficient resources that I could say yes more often than I said no, an ideal position to be in. And we made great progress during that time under the Bienen administration. Now, while at Northwestern, I had tried to recruit Susan Lindquist from the University of Chicago to Northwestern. At the same time, Sue was being courted to come to the Whitehead Institute here in Cambridge. After she got here, she recruited me to become the COO and Vice President for Research at the Whitehead. And while I was there, the main job was to move the Genome Research Center, which was a Whitehead activity, to an independent center affiliated with MIT, Harvard, and the Whitehead. After 3 years, Susan decided that her heart and mind were focused on her scientific work, and she returned to her laboratory as a Howard Hughes investigator. I left Whitehead and joined Cytonome. That company had been started by John Gilbert to develop the instruments you heard about to sort human cells very rapidly under sterile conditions. My intent was to stay for a year, but I stayed 8. John left to start other companies, and I became CEO and had to find money in 2008, just as the economy was coming to a crash. During that time, the executive team took no salary. I had to lay off half the staff, and I made over 200 pitches to no avail. It was much harder than my time at Cold Spring Harbor. Then, in 2009, an opportunity arose from a most unexpected source. A company based in Texas, now called Genetics ST, was using conventional cell sorters to produce bovine sperm enriched for the X chromosome. This sperm was sold to dairy farmers. because dairy farmers have to replace their milk cows about once every 3 years. They don't have so much use for the male calves. So this company was on the rise. The CEO of that company, Juan Moreno, liked our technology, and so he decided to invest, and John Sharp, who had vast experience building cell sorters, took over as CEO. I stayed on as CSO and on the board until 2014 when I left the company but not the board. At present, Cytonome still supplies Genetics ST with sorters, and they are now spread all over the world, working 24/7. And the Gigasort is in use in Japan for a clinical trial conducted by Sumitomo Pharmaceuticals to sort differentiated neuronal cells to treat Parkinson's disease. We think that a trial will start in this country soon. After I left Cytonome, it took me a little while to decide how I wanted to spend my time. Ultimately, I decided to start a one-person consulting company to serve as an umbrella for my activities. I had always been puzzled by how slowly women and people from historically marginalized groups were incorporated into science, technology, engineering, math, and medicine. Many people I knew in these fields seemed to agree that more diversity was needed and desirable. Well, a friend of mine, Florence Bonner, who was VPR at Howard University and a social scientist, said to me, you know, you natural scientists tend to talk about diversifying STEM, but you aren't considering any of the things that we, the social scientists, have learned. That made sense. So I began to look into studies on human decision-making, including the role and neurological underpinnings of unconscious bias. Bottom line, unconscious biases are universal. All of us have them. It's a big data problem. Our minds, as wonderful and as complex as they are, has to have shortcuts because it cannot deal with the amount of data we are receiving all the time from all our senses. I developed a formal presentation when the program on science, technology, and society at MIT awarded me the Elting E. Morrison Prize in 2016. My role now is to provide a dispassionate, data-driven explanation for why well-meaning people have not been able to improve the diversity of STEM fields. My goal is to open minds, especially at the decision-making level. One lecture will not change any minds, but it might make it easier when someone else at the institution suggests some processes to ensure that implicit biases mean that individuals are not eliminated before their credentials are evaluated. As of this writing, I've given about 80 or so presentations to universities, colleges, companies, and federal agencies. My degree is in cell and molecular biology, and it's enabled a career, though, in many different fields, in a whole bunch of roles. That's what you've gotten with your degree, the ability to learn, to think critically, to be persistent. The specifics of your study will become less important as you proceed through your career. In other words, your degrees are not degrees that define limits. It is a key to doors, and which doors you go through are up to you. Thank you, and congratulations once more.
Thank you very much, Lydia. That was a fascinating story and a trajectory and a path. And now, on to the reason we're all here today, our graduates. So here is some official language. I'll try to get it right. The faculty of the School of Science are declared convened. I hope you feel convened. Together with this assembly for the conferring of master's and doctoral degrees, including the investiture of the doctoral academic hood. The academic hooding ceremony is a symbol of dedication to scholarship and attainment of the doctoral requirements defined by the Academy. I am proud to now introduce you to MIT's newest scholars.
Course 5, Chemistry, Master of Science in Chemistry.
So Yi Chung. Nityakan Kuichad. Brittany Lynn.
Course 12, Department of Earth, Atmospheric and Planetary Sciences, Earth and Planetary Sciences.
Vittorio Calici. Neosha Gupta Narayanan.
Master of Science in Chemical Oceanography, jointly by Massachusetts Institute of Technology and Woods Hole Oceanographic Institution.
Erica Lauren Herrera.
Master of Science in Physical Oceanography, jointly by Massachusetts Institute of Technology and Woods Hole Oceanographic Institution.
Helena Cheslak. Oakland Keefe.
Investing hoods for the Doctor of Biology on behalf of the Department of Biology, Professor Mary Gehring, David Baltimore Chair in Biomedical Research.
Linda Zong Johnson. Jamilex Acevedo Sanchez. Swanee Lamboy Rodriguez.
Luis Martinez Rodriguez.
Connor Lee McMahon. Arish Shah. Chan Young Park. Barrett Powell. Jade Verino. Nicole Louise Fendler. Victoria Hernandez. Michael Doss. Laurel Kinman. Kathleen Whitmore Higgins. Avinoam Singer. David Driscoll. Colin Fowler. Sarah Chang. Anna Ye. Catherine Koch. Juliet Barker. Irene Zhang. Annika Gomez. Jade Bath. Veda Kadka. Jacob Baker. Kristen Andrukovich Fing. Martin Ariola Villanueva.
Investing Hoods for the Doctor of Philosophy on behalf of the Department of Brain and Cognitive Sciences, Professor Mriganka Sur, Director of the Simon Center for the Social Brain.
Dae-hee Yoon. Michael Alexander Skahersky. Andrew Bailey. Iqbal Hosseini Asl. Karina Kauf. Guren Kar Madan. John Tauber. Gabrielle Drummond. Catherine Zimring.
Sarah Kornfeld Simpson.
Alexandra Ferguson. Mahdi Ramadan. Victoria Beha Glasser.
Investing hoods for the Doctor of Philosophy on behalf of the Department of Chemistry, Professor Elizabeth Nolan, Associate Department Head, and Ivan R. Cottrell, Professor of Immunology.
Giselle Andre. Landon Kilgallen. Yi Fan Wu. Xin Gu. Haojun Jia. Michael Mardini. Samuel Kaiser. Xu Ming Lu. Allison Beester. Amanda Kaufer. Michael Lee. Ardavan Faravash. Maria Alejandra Castellanos. Jet Lem. Blake Dastrup. Noah Benjamin Lewis. An Chu. Trevor Bostelar. Kimberly Hollister. Janet Pete. Hadika Zafar. Leticia Cardoso da Costa. David Berkinski. Atharva Sahasrabuddhe. Jing Yi Zhen. Deepxing Shangtan. Vaishnavi Venelakanti Veluru. Catherine Marie Yamin. Catherine Walker. Jolyn Giancola. Evans Ralsted. Eric Sung. Chu Chu Guo. Jin Yi Yang. Brian Tong. Gil Namkung. Wei Lun To. Jessica Beard. Brighton Alexander Skeel. Kayla Storm. Stephanie Michelle Amtree.
Investing hoods for the Doctor of Philosophy on behalf of the Department of Earth, Atmospheric, and Planetary Sciences, Professor Rob Vanderhilt, Department Head and Schlumberger Professor of Earth and Planetary Science.
Diana Dumit. Mariona Badenas Agusti. James Hall. Elias Nathaniel Mansback. Susana Ojos Munoz. Anna Rose Walsh Glidden. Chris Parsons. Zachary Molitor. Martin Velez Pardo. Ali Ramadan.
Investing hoods for the Doctor of Philosophy on behalf of Mathematics, Professor Michel Gomans, Department Head and RSA Professor of Mathematics.
Julius Baldauf. Murillo Corato Zanarea. Danielle Wong. Gefei Dong. George Stepanyants. Adam Block. Gerber Patrick Robert. Calder Morton Ferguson. Matthew Nicoletti. Catherine Cawley Wolfram. Pu Yu. Alexander Ortiz. Ashwin Sa. Arun Srinath Kanan. Chen Lu. Felipe Suarez Colmenares. Shashi Gowda. Alistair Hastewell. Jae-hee Lee.
Investing hoods for the Doctor of Philosophy on behalf of the Department of Physics, Professor Deepto Chakrabarty, Department Head.
Mason Ng Wei Tie. Jing Yi Wang. Dhruv Ganapati. Benjamin Lane. Michael Calzadilla. Stephanie O'Neill. Danielle Frostig. Lisa Drummond. Jong Min Jane Park. Wenzer Chin. Yi Tian Sun. Sarah Geller. Zane Marius Rossi. Annie Wei. Zhihuan Dong. Andrew Tan. Nicholas Romeo. Osman Shagar Folkestad. Robert Johnston. Patrick Moran. Adam Treback. Li Du.
Investing hoods for the Doctor of Philosophy on behalf of MIT and the Woods Hole Oceanographic Institute, Professor Mick Follows, MIT Director of the MIT-HUI Joint Program in Oceanography.
Samantha Clevenger. Jordan Pitt. Corey Berger. Donald Marticello. Paris Smalls.
To conclude the investiture of the doctoral hoods, I was supposed to invite the faculty to join me in saluting a welcome to you, the class of 2024, but I think we jumped the gun. So again, a warm congratulations to you, our doctoral degree recipients, and also our master's degree recipients for your outstanding academic achievement. With your graduation today, the faculty of this institution recognize the achievement of our advanced degree candidates. Congratulations to you all. Now, almost as much hard work as earning your degrees, I also want to give a round of applause and thanks to the dedicated staff who've helped put together today's advanced degree ceremony. Thank you all. So, family and friends, I ask that you remain in your seats until our faculty and graduates have exited to the lobby. Once they're out, please join us for a reception just outside in the tent. Again, congratulations. Thank you all for being here, and more congratulations.